Audio check. Okay. All right, genetics. Hope everyone's doing well. We're getting into chapter 3 here. So chapter 3, definitely have it broken up into 2 parts. So this is Mendelian genetics, the basic principles of heredity. And basically with the first part, we're going to get into what Mendel used as his model organism, and we'll do a monohybrid cross. We'll get into back crosses and test crosses, which are very important in terms of calculating probabilities. I'll also introduce you to some rules of probability. And then in the second part of chapter 3, which will be a separate lecture, we're going to do the dihybrid cross, and we'll look into how to calculate chi-squared values as well. Okay. So again, I just want to reinforce this is a really important chapter in genetics. The next 3 chapters are very crucial. This one especially, because this is where it all starts. You want to get these foundations down perfectly. Okay, you want to be able to teach this to someone else. That's how well you know it. Because if you can do that, for the most part, you should be able to answer most of the questions that are given to you. Okay. All right. Let's get going here. So Gregor Mendel, which is considered the father of genetics, had great success. Why was he so successful? Well, he picked a great experimental model, the pea plant, and he used statistics, right? So he analyzed all the results mathematically, and with statistical power, now you can prove you can prove the results that he got. Right? So a little bit about the pea plant. What was so perfect about the pea plant is that it's easy to grow, it's quick to grow, and it has these very apparent characteristics like seed color, seed shape, pod color, pod shape, how the flowers are positioned along the stem. Are they positioned in an axial orientation, which means up around the stem, or was it terminal, which means that they're at the top of the stem? Or the plant could be short or tall, right? So he had these certain phenotypes, and then for each phenotype, he really only had 2 different possibilities. Okay, and there's some others. These aren't all of them. These are the more commonly encountered ones. The flower color, I think, should be on here. Well, that's not. So they only had 2 versions, right? So there were only 2 alleles for each phenotype. Okay. So as you work through these, you'll become more and more familiar with which allele is dominant and which allele is recessive, and we'll talk about what those mean. But just to kind of give you a spoiler alert here, typically the letter for the abbreviation, how it's abbreviated in the problem, it takes the first letter of the dominant allele. So this phenotype right here would be dominant over green, and that's why we say it's Y, is the allele for that seed color, whereas little y would represent green. Okay, so typically the first letter of the dominant allele is the letter that's used for both alleles. All right, so with dominant, we say it's uppercase, and with recessive, we use the lowercase letter. Okay, and he was able to prove dominance and recessiveness through his experiments. But basically, if you have an individual plant that has, for his homologous chromosomes, he has the yellow allele on one chromosome and the green allele on another chromosome, well, all his seeds are going to be yellow because yellow is dominant to green. Okay. All right, which of the following factors did not contribute to Mendel's success in his study of heredity? His use of the pea plant, his study of plant chromosomes, adoption of an experimental approach, his use of mathematics. So hopefully you have answered chromosomes, because at the time Mendel knew nothing of chromosomes. But the use of the pea plant, experimental approach, and his use of statistics was certainly vital to his success. Okay, important terms, right? So a gene is something encoded in the DNA, and it helps determine a certain characteristic, right? This is an inherited factor. An allele is one or two or more, but in Mendel's cases, which is nice, it's just two, alternative forms of a gene, right? So for one gene, you have multiple alleles, okay? The locus is the specific place on the chromosome where the allele is located. Pretty easy to remember that, locus, located. And a genotype is a set of alleles that an individual has, right? So it's the actual like DNA code, whereas a phenotype is the appearance or the manifestation of that set of alleles, essentially, or the characteristic, okay? Individuals that are heterozygous means they have two different alleles at a locus on two chromosomes, on a pair of homologs. If they're homozygous, the same allele is present at a particular locus on two chromosomes, which are homologs. Okay? All right. Be sure you understand these. The more you work through this and read it, it's going to become more clear. It'll become like second nature. You just have to put in the work to do that. All right. So here's like a pair of homologous chromosomes. So here's a locus on each chromosome. And this, whatever this gene is, what is this? Okay, round and wrinkled, right? So round is the dominant, and that's why we use the letter R to indicate this. So, and then lowercase r, which is the recessive allele, is found on the other homologous chromosome, right? But same location on the chromosome, because remember, these are the same chromosomes, essentially. One's from mom, one's from dad. It's the same chromosome number, whatever, and it has the same genes on it. There's just different versions of it because of, things like crossing over and genetic recombination, all those things that give variation. What is the difference between a locus and an allele? What is the difference between a genotype and a phenotype? Well, a locus is a location on the chromosome. An allele is a variant of a gene, right? So different versions of genes on different chromosomes. What is the difference between genotype and phenotype? Genotype is the really actual DNA code, right? The set of alleles, the genes. Phenotype is the physical manifestation. So what you can visibly see or detect. Doesn't have to be something you can see. Blood pressure is a phenotype, right? You can't see that. I guess if it's real bad, maybe you could. But okay. Okay, so the first thing Mendel started to do was do these things called monohybrid cross. Let's look at that word monohybrid. Mono means one. And what does the word hybrid mean? Well, a hybrid is when you cross two parents that are true breeding. I don't know why I don't have that term on here, but it's important. True breeding. So what does true breeding mean? Well, so one thing with Mendel's pea plants is you could have them fertilize themselves. And that was very important in these experiments, right? So it's a simple surgery where you just take like a paintbrush and you pull some pollen off of the plant's flower, and then you just stick it in the other reproductive organ, right? And then it will fertilize itself. So you can maintain those genetics. So a true breeding plant is a plant that when mated with itself, 100% of the progeny, 100% of the time, are going to be identical to the parent, right? So basically you've gotten rid of all of those recessive genes. He's dominant for everything. Every time you mate it with itself, the progeny are identical to the parent. Okay. So what Mendel did to do his monohybrid cross is he took two true breeding parents that were different for the same gene, had different alleles. For example, right, he took a parent that was true breeding for yellow and a parent that was true breeding for green, and he crossed them together. And what does that mean if they're true breeding? Well, this one, when you self-fertilize it every time, they're always yellow. No except, without any exception. And this guy, true breeding for green, when you self-fertilize it, 100% of the time, all the seed color is going to be green. So in monohybrid crosses, because we're hybridizing, that means different, right? So we're taking two different phenotypes, but each of them are true breeding, crossing them with each other, and now looking at the progeny. So what did he do? Well, so here's what I'm talking about with this. Now, this isn't a self-fertilization, or he is self-fertilization. Cross different varieties of peas, Mendel remove the anthers from flowers to prevent self-fertilization. Right. So he didn't want it, if it's self-fertilized, then it would mess up his results. This is how he did a cross. But because he was able to do this, he could also generate true breeding plants. Okay. Anyway, so this is the monohybrid cross. So what he's doing here is the parental generation is homozygous for round seeds and homozygous for wrinkled seeds. So he crosses them, and we abbreviate these generations P, F1, and F2, right? So P stands for parental, F stands for filial, and filial, that's the first filial generation, and then this is the second. Okay, so he crossed them, and he looked at the F1 generation, and he saw that all of the seeds were round, right? So that wrinkled phenotype didn't carry through to the F1 generation. That's why, and this kind of, this came him to realize that some traits are dominant over others, right? So round is dominant to wrinkled. That's why when we do abbreviations with this, which you need to start getting used to, we're going to use the letter R for round and wrinkled, and little r is going to represent wrinkled. Now, in monohybrid crosses, and actually all his crosses are always a cross of the parental to give the F1 generation, and then a self-fertilization of the F1 generation. That's the direction, that's the flow of how these crosses always go. Okay, cross of parental, self-fertilization of F1, and then you evaluate the F2. When he did a monohybrid cross, what he found was that 3/4 of the peas were dominant or round, and 1/4 were wrinkled. All right, so now what kind of ratio is this? Well, this is a phenotypic ratio because we're visibly looking at the seeds. 75% are round, 25% are wrinkled. Okay. So what did he conclude? Well, there's no blending of traits, right? The traits stay on course, right? This, you have only round here in the F1 generation, and then in the F2, you have round and wrinkled. There's no blending together. You don't have ones that are like partially wrinkled or something like that. Oops, sorry. Okay, important ratio, 3-to-1 phenotypic ratio. You have to memorize that. Be able to do calculations. Just want to check something. Is this getting into the genotypic ratio? Here goes the genotype. Okay. All right. So monohybrid crosses, what do they reveal? Well, they reveal the principle of segregation. This is Mendel's first law. Right? If you understand meiosis, you can understand the principle of segregation because it's happening during meiosis. So each individual diploid organism has 2 alleles for any particular characteristic. Okay? And one of those alleles could be dominant or recessive. The other allele could be dominant or recessive. These alleles segregate when gametes are formed, and one allele goes to each gamete, right? The homologs separate during anaphase 1. The chromatids separate during anaphase 2. And also the concept of dominance. So when two alleles are present, right, only the trait encoded by one of them, which is the dominant allele, is actually observed in the phenotype. Okay, so yes, concept of dominance and principles of segregation, which were proven by the monohybrid cross. Now he has an additional law, right? The law of independent assortment, and that is proven in a dihybrid cross, right? So 2 out of the 3 laws that Mendel came up with got proven just with a monohybrid cross. Okay, so these monohybrid crosses are explained by the principle of segregation, right? And it's important we get used to using these symbols of genetic crosses. So now let's apply nomenclature in the form of letters to these phenotypes. So we're going to say this here is homozygous dominant. Homozygous because this individual who is true breeding for round seeds, right, has the round seed allele on both chromosomes of the homologous pair. Okay, this individual has the recessive allele on both chromosomes of the homologous pair. So we say he is homozygous recessive. Homozygous dominant, homozygous recessive. So after meiosis, when the gametes form, all of the gametes in a person that's true breeding are going to be the same as the parent because they have the same allele, right? So 100% of the gametes are going to be big R. 100% of the gametes over here are going to be little r. During fertilization, the big R and the little r come together, and that's why you have heterozygotes. Okay, homozygous is when the alleles are the same. Heterozygous is when you have one dominant and one recessive allele. When you do a monohybrid cross, the F1 generation is always 100% heterozygous. And then look how these gametes come apart during meiosis. So you will have 50% that have big R and 50% that have little r. Okay. Remember, you're doing a self-fertilization for the F1. So we are combining 2 heterozygote organisms, big R, little r, combined with big R, little r. All right, now there is a very much easier way to figure this out, and it's called a Punnett square. I don't know why I don't have one drawn here. I have one here. I'm going to jump ahead of this slide real quick because it's just easier to visualize the math, especially when these get more complex. But you are combining these together, right? So you could also look at it this way. Big R combines with big R here. There's your homozygous dominant. Big R combines with little r here. Here is your heterozygous. This little r combines with this big R. Here's your other heterozygous. And this little r combines with this little r. There's your homozygous recessive. Okay. All these are the results of self-fertilization. So you have basically, now this can be misleading because someone sees this and they're like, well, there's 1, 2, 3, 4 different genotypes here and 3 different phenotypes. Okay, that's not correct. There's only 2 different phenotypes, round and wrinkled. And there's only 3 different genotypes: homozygous dominant, homozygous recessive, and heterozygous. Okay, these are not different genotypes. You would just indicate this big R, little r being 50% of all of this. Okay, so an easier way to get this math here is to do a Punnett square. Punnett square is very easy. You've probably seen this before, where you just take the gametes of one organism versus the gametes of the other organism, and you bring them together. Right, so this one is homozygous recessive, this one is heterozygous, And this comes down here and this goes here. And rewrite it. This goes here and this goes here. It allows us to easily distinguish the genotype and the phenotype of the progeny. And you're going to do a couple hundred of these in this course. And as you get going on them, you can start to do them in your head. But as a general rule, I would just write it out. You're allowed scratch paper on these on the exam. You have to show it to me when you start it, but you are permitted scratch paper because you're going to need it when you're working these problems. Okay. All right. So F3 generation. Well, honestly, we don't really look at F3 generations typically, except when we're doing pedigrees. Mendelian genetics, though, really just take up to the F2 generation. Okay. I mean, he did do F3 generation evaluation where he took the heterozygotes and he would do self-fertilization, and you could see, the recessive alleles still coming through, right? So, all right, perform a monohybrid cross between two true breeding parents, one tall and one short. Find the results in genotypic and phenotypic ratios for the F2. Okay, so like I said, these lectures correspond to like my in-class thing. It's hard for me to, I want you guys to take a second and do this, but I don't know if I'm going to be able to, actually show you the results. Let me check something here. Hold on. I have this. I love tech, guys. I really just hate tech. Hold on. I have this drawing pad, but I don't know if I never use it. I think it works. Yeah, it doesn't even work. Yeah, I don't know. This thing's not set up. Paper made. Oh, that was an actual pen that I was using. Okay, I'm sorry. All right, let's see if I can use this pen here to. Oh my God. Let's see. Great. Huh. Okay. Perform a monohybrid cross between two true breeding plants, one tall and one short. So that means we're going to cross big T versus little t, right? So this is the parental generation. We're doing a homozygous dominant versus a homozygous recessive. The F1 generation, when we cross these, are going to be 100% heterozygous, big T, little t. So this is 100% tall. That's the genotypic, and 100%, big T, little t. Okay. For the F2 generation, well, we're going to cross this with itself. Let's do a little Punnett square over here on the side. Big T, little t, big T, little t. So that's going to make big T, big T, big T, little t, big T, little t, and little t, little t. Okay. So the F2 generation is going to be, so this right here represents 75% tall and 25% short. Okay, that is the phenotypic ratio. All right, now for this one, F1, I gave the phenotypic ratio 100% tall. And I also gave the genotypic ratio, which is easy, right? It's 100% heterozygous. Now for the F2 generation, the genotypic ratio is a little different. So because we have homozygous dominant, we've got homozygous recessive, and we also have heterozygous. So the genotypic ratio here is 1. I don't know why it's doing this stupid thing. 1 to 2. Oh my God. It's 1 to 2 to 1. So that was working there for a second. 1. Okay. Well, anyways, I'm not going to bore you with this. So the phenotypic ratio is 3 to 1, right? 3 tall to 1. So 1 out of 4 is short. The genotypic ratio is 1 to 2 to 1, right? So it's a total out of 4. Phenotypic ratio 1 to 2. All right, so why are these ratios important? Well, no matter what trait or characteristic Mendel looked at, as long as he crossed 2 true breeding parents, and as long as he self-fertilized the F1 generation, he always got these ratios. He always got a 3 to 1 phenotypic and a 1 to 2 to 1 genotypic ratio. And it's understanding this that is crucial for being able to answer these questions. Get on the exam. Okay. How did Mendel know that each of his pea plants carry 2 alleles encoding a characteristic? Well, they both appeared in the F2 progeny, right? So he didn't see one of them. He didn't see the recessive allele in the F1, but he was smart enough to keep going, self-fertilizing, to keep searching for these traits, and think that because he didn't see any blending, so he did see it eventually in the F2 generation. Okay. All right. So so that's the law of dominance, right? This question has to do with like the law of dominance, and then what this is talking about the law of segregation, right? So the separation of the chromosomes during anaphase. So just a reminder about meiosis, right? So here we've got a cell that's becoming a gamete, right? So it's becoming a sex cell, and it has a homologous pair right here. And on one chromosome, you have one allele, and the other chromosome, you have the other, right? So this is big R, this is little r, right? During S phase, everything duplicates. Now we're big R, we're little r, right? So remember, this is still one chromosome, right? So we still got 2 chromosomes in here only. We have 4 chromatids. Now, there's 2 things that can occur, right? You can have crossing over or you don't. All right. It doesn't matter though. I mean, if you do, you're still going to end up with 4 gametes, right? You're still going to have segregation. But in the case with crossing over, you will get increased variation, right? Really, the importance though of this figure, it's just trying to show you the law of segregation, which is that during anaphase 1, the homologs separate, and during anaphase 2, the chromatids separate. That's all it's trying to show you. Okay, so how can you do tests, right? So how can you test for individuals that you don't know, like the phenotype or the genotype, okay? Well, you can start to do what are called backcrosses, okay? So a backcross is a general type of cross because there's different types of backcrosses, but the most useful type is something called a test cross. So a test cross is actually a type of backcross. A backcross is when you cross any F1 genotype with either of the parents, okay? So you can test any F1 genotype, right? Which in terms of monohybrid crosses are always going to be 100% heterozygous with either the homozygous dominant or the homozygous recessive, okay? Here's an example of a test cross and also shows the Punnett square. So in this test cross, I'm sorry, backcross, I haven't used the word test cross yet. In this backcross, we are testing, here's the F1 generation, okay? And here is the homozygous recessive of the parent. Okay, this is one test cross. We've already done our monohybrid cross over here. That's what this is assuming. And our test cross gives our results. And you can see they are 50%, right, heterozygous and 50% homozygous recessive. So a 1-to-1 genotypic ratio and also a 1-to-1 phenotypic ratio. Okay, so when you start doing different crosses, now you start getting different ratios. But with monohybrids, whenever you do two true breeding plants that are differing in only one characteristic, you always get a 1 to 2-to-1 genotypic ratio and a 3-to-1 phenotypic ratio, okay? All right, so here is test cross stuff. Here's the test cross. There's this test cross. Sorry, just bear with me a sec. There it is. Okay, I want to make sure we're getting to that in this chapter. All right, backcross is backcross. If an F1 plant depicted in figure 3b, so I went ahead and took 3b and pasted it on this slide, is backcross to the parent with round seeds. A typical F1 plant is backcross to a parent with round seeds. So that means big R, little r, crossed with big R, big R. What proportion of the progeny will have wrinkled seeds? Well, if I can get this thing to work. So if we're crossing big R with one of the F1s, right? So this is our progeny. It's going to be big R, big R, big R, big R, big R, little r, big R, little r. 100% of our progeny is round, right? So even though 50% is homozygous dominant, 50% is heterozygous, because heterozygotes are the round, 100% are round. Wrinkled is homozygous recessive, and we don't get any when we do that test cross, okay? That back cross. Okay. All right, so the answer there is 0. Okay, so now that is a very tedious way of doing mathematics, and there are better ways of doing like calculations, and you're going to have to learn to do it. You can't just do, like when we get to dihybrid crosses, you can't just draw out a Punnett square for most of the calculations. Okay, you're going to have to use like a binomial expansion to do it. So we're going to learn 2 very easy rules here that, again, you have to have knowledge of like the back of your hand, right? But they're very simple. So let's use a rolling a die as an example. If you take a die and you roll it, and let's say we're trying to get the number, 4. We're trying to get the number 4. So if you roll it, well, there is a 1 out of 6 probability that you're going to hit 4, right? You don't have to spend a lot of time in Vegas to understand that. It's a 6-sided die, so there's a 1 in 6 probability that you will hit a 4. All right, now, if we have a 1 in 4 probability or 1/4 probability of hitting a 4, what is the probability of hitting it again? Okay, and that's where this multiplication rule comes into play. And the key word in knowing to use the multiplication rule is the word end. What is the probability of getting a 4 and another 4? Well, it's 1/6 times 1/6 equals 1/36. Pretty easy. equals 1 over 36. Sorry. All right, so the word and, you see the word and, think multiply. If you see the words either or, think add. So if you roll a die and on average, 1 out of 6 times you get a 3, right? And then 1 out of 6 times you get a 4. What is the probability of getting a 3 or a 4 is, well, you use the addition rule. You add 1/6 plus 1/6, and you get 1/3. Okay, 1/6 plus 1/6. So addition, when you have the words either or, and multiplication when you have and. Okay. So assume we cross, let's look at this conditional probability example. Assume we cross 2 heterozygotes, okay, big T little t and big T little t, and we obtain a tall plant. What is the probability that this tall plant is heterozygous? Okay, now wait a second, think about it. So this is typical Mendelian monohybrid cross. This is like what you do with the F1 generation to generate the F2. So we know that the F2 generation is going to have a 3-to-1 phenotypic ratio. 75% or 3 out of 4 are going to be tall, 25% or 1 out of 4 are going to be short. And it's got a 1-to-2-to-1 genotypic ratio. So let's write this stuff out. Here's our genotypes, 1-to-2-to-1. 1/4 are going to be homozygous dominant, 1/4 are going to be heterozygous, and 1/2 are going to be, I'm sorry, I said that wrong. 1/4 are homozygous dominant, 1/4 are homozygous recessive, and 1/2 are 50% are heterozygous. Now, what is the probability that this plant, what we obtain a tall plant? And then what is the probability that tall plant is heterozygous? Well, only these have tall or make tall plants. So only these genotypes are tall. This genotype is short, so we can eliminate because we know the plant is already tall. What is the probability that this tall plant is heterozygous? So 1/2 of this entire whole, right? Okay, so 50% of 3/4 is what you're calculating here. And the probability that this tall plant is heterozygous is going to be 1/3. Right? Heterozygous. Oh, it's 2/3. Sorry. Probability that it's homozygous dominant would be 1/3. Okay. So 2/3, 1/3 homozygous dominant. So there's something missing there. Oh, okay. If the probability of being blood type A is 1/8 and the probability of blood type O is 1/2, what is the probability of being either blood type A or O, right? So, we're doing either or, so we're going to use the addition rule. So we're going to add 1/8 and 1/2, and that gives you 5/8, right? Yes. Okay. So, so if you've forgotten how to add fractions, which happens because people don't do it too often. I'm not going to tell you how to do that. Just look it up, get a YouTube thing. Okay. You have to find the common denominator. But adding 1/8, right, 1/2 gives you 5/8. Okay. All right. Okay, so we talked about back crosses. That's when you cross one of the F1s with either parent. The test cross is like a particular type of back cross where one individual where you don't know the genotype is crossed specifically with homozygous recessive. And this is always going to be your most useful type of cross, right? Because the recessive trait doesn't have like all that penetrance, right? It doesn't come through unless it's homozygous. So this test cross is very valuable. Let's do an example of a test cross. All right, so here is this. Oh, I was trying to figure out where is this. Sorry, guys, just give me a sec here. There. All right, so let's do an example of a test cross. You have progeny where all the plants are tall. All right, let's figure out their genotypes. So you would know that the plant, right, if it's tall, it could be either big T, or big T, little t, but you don't know which one. So you could do a test cross to figure that out. So we're going to take each of these genotypes and we're going to cross it with the homozygous recessive. When you cross big T times little t, when you cross homozygous dominant versus homozygous recessive, you get 100% tall, 100% heterozygous. When you cross heterozygous versus homozygous recessive, you get 50% tall, 50% short, because 50% heterozygous and 50% are little t, little t. Right? So, what are the genotypes? Well, the plants would have to be heterozygous, right? Otherwise, it would be a combination of both combination of both heterozygous and homozygous recessive. So here is your test cross, because you're crossing these unknown genotypes with the homozygous recessive. Okay, so before we do the second part, genetic symbols, right? So you're going to see this stuff. Now, when we get into the next chapter, we're going to start using like the term wild type a lot more often because we're going to be doing Drosophila. So wild type is denotes the normal phenotype, right? So wild type typically uses a plus sign, and if you're denoting a mutant phenotype, it will use letters though. So like just a plus sign alone, right, indicates like the wild type. All right, you'll see superscripts and subscripts. Okay, these represent like dominant alleles at different loci. All right, and this is a very important one. The underscore indicates that any allele is possible and it's unknown. Right? So usually you're not necessarily calculating for that, but it could be when you're doing your calculation, this could be either heterozygous or it could be homozygous dominant. You would just have to do both calculations, right? Because you don't know that's that other allele. All right, guys, that is it for the first part of chapter 3. Hopefully you guys, it's really important with this chapter to work the problems at the end of the chapter. I will get the second part put up hopefully tonight. My kid's got a baseball tournament this weekend, and I got to drive up there tonight. Hopefully at the hotel I can get the second half of the chapter posted for you tonight, okay? All right, you guys keep studying hard, and I will be in touch here